Welcome to our experiment on the effects of varying pH levels on mung bean germination. We'll examine how varying pH levels impact the germination of mung bean seeds. pH scale is used to measure how acidic or alkaline a substance, usually a liquid, is. The range is 0 to 14. 0 being very acidic as found in highly concentrated hydrochloric acid. This is very dangerous. On the opposite side is alkalinity, which goes to a pH of 14. An example of this is sodium hydroxide, which is used for cleaning drains and is also very dangerous if mishandled. In the middle of this scale is pure water, which has a pH of 7. This is considered neutral pH. In food, lemon juice is acidic and has a pH of around 2. And on the other side are alkaline foods such as leafy greens like spinach, kale, and Brussels sprouts that can have a pH of around 9 in some cases. We can safely handle the pH range of 2 to 9 for this experiment without requiring extensive protective equipment. Germination in plants is the process by which a dormant seed begins to sprout and grow into a seedling or baby plant under proper conditions. The basic growing conditions usually involve water, a range of temperatures, not too hot or too cold, a material to grow the plant, sometimes called a media, don't call it dirt, and eventually light. Now back to our experiment on the effects of varying pH levels on mung bean seed germination. These tiny seeds have the potential to grow into healthy bean sprouts. But what happens when we expose them to different pH environments? Let's find out. We've prepared pH samples ranging from 2, which is acidic, to 9, that is alkaline. We'll use these solutions to create different environments for our mung bean seeds. Now we'll place 50 mung beans onto filter paper in each black grow dish, carefully moistening the seeds with their respective pH solutions, ensuring they're soaked but not submerged. These seeds are all set in their respective pH environments. Now we'll cover the black grow dishes to maintain moisture and place them in a controlled environment with consistent temperature and light. Over the course of 3 days, we've observed some interesting developments. Let's take a closer look at the results. As you can see, at the two extremes, when you have a pH of 9, the seeds have gotten very dark and none of them have sprouted. On the other side, when you have an acidic pH of 2 and 3, there was no sprouting. These seeds look like they were cooked. Now is the time for you to do some work. I've collected the data for you to determine the optimal pH level for germinating mung beans. As you can see, growing food involves math and science. In this case, statistics and chemistry. This is applied to plant biology in determining the optimal germination pH. Thank you for joining us in this experiment. Understanding how pH affects seed germination can guide us in creating favorable conditions for plant growth and crop production.